Post-decompression bubble and inflammation interactions in divers. Despite its low incidence, decompressive disease, or the bends, is a great concern among divers. It can cause weakness, pain in muscles and joints, stroke, and until death. Symptoms occur because, in the ascent of the dive, the accumulated nitrogen can form microbubbles in tissues, blood and joints. To prevent the DCS, divers take breaks during the ascent, called decompression stops, so that the gas diffuses out of the tissues and is released by breathing. In addition to bubbling, it is believed that decompressive disease activates the immune system, triggering a potent inflammatory process. To verify if the number of decompression stops interfere with the formation of bubbles, the experiments were divided into two profiles: ZHL profile with 3 stops and ZHL adjust profile with 8 stops. All volunteers used autonomous respiratory system (SCUBA). The 2 dive profiles, which we will show next. In the experimental tests, all volunteers descended to a pressure of 528 kPa, and remained at the bottom for 40 minutes. The percent gas composition at the first stop is 21 oxygen, 35 helium and 44 nitrogen. The ZHL profile, a decompression rate for the second stop, and the respiratory gas was changed to 50 oxygen and 50 nitrogen. At the third stop, the composition of the gases remains. In the adjusted ZHL, the ascent pattern was identical with the same gas composition. However, in this profile the number of decompression stops was increased. The researchers from the Laboratory of Energetic and Theoretical Physiology, Department of Physiology of the University of São Paulo, studied some factors related to the formation of bubbles and inflammatory processes during dive decompression to find the best way to represent this relationship. They identified that the formation of beta is initially triggered by nitrogen pressure ρ rising above barometric pressure. This formation of bubbles causes the increase of a pro-inflammatory factor, psi, The pro-inflammatory factor can also be stimulated by inert gas pressure. The simultaneous presence of bubbles and pro-inflammatory factor stimulates an inflammatory factor, zeta. The inflammatory factor also stimulates the production of the pro-inflammatory factor. This interaction between beta, zeta, and psi gives two important characteristics for the model. One is a self-sustaining process, and two may be aborted by a recompression procedure. Thus, the differential equations of each of the variables were formulated. Comparing the experimental data obtained by Doppler with the simulations of the mathematical model, this research identified the same behavior in the formation of the bubble profile. And the average formation of bubbles over time, arbitrary time, of greater degree in the ZHL profile than in the adjusted ZHL profile. That is, the profile adjusted with more dive stops presents in this study less bubble formation. The main objective of this work is to model the formation of bubbles and inflammation in decompression diving procedures. The model clearly captures this interaction. The model shows that a relevant increase in the formation of bubbles that occurs concomitantly with the inflammatory process triggered by decompression. Another interesting point presented by the model is the contribution of different compartments to the overall process detected at the body level. Example: bubble counting. Fast compartments such as CNS, kidneys, and heart would have a low contribution to the process as a whole, while intermediate compartments such as skeletal muscles and gastrointestinal tract would be the main sites of bubbles, inflammation after immersion. Slow compartments, such as adipose tissue, may go unnoticed since the dynamics of gas is delayed in these tissues during diving, decompression. These results are obvious in the sense of gas dynamics. What is new is that inflammation becomes potentially exported from one compartment to another even without arterial embolism, resonating potential for DCS.